Good morning, everyone. Today we're going to be talking about invertebrates. This is the first PowerPoint, so here we go. Now, just some things to know about animals in general, whether they're vertebrates or invertebrates. They are in the domain Eukarya, so they have nucleus and they have organelles, and they are also in the kingdom Animalia, obviously. Some characteristics that you would need to know: they are heterotrophic. They get their food by ingesting it from outside sources, and they also ingest it on the inside of their bodies. They're usually mobile, which means they can move by means of muscle fibers. They are multicellular. They have high tissue and cell specialization that helps them form organs and tissues and organ systems. The adults tend to have a full number of chromosomes, so they are diploid, and reproduction is usually sexual, and the embryo is going to undergo developmental stages. They tend to have a longer gestation period than, say, bacteria or fungi or something like that. Now, invertebrates, obviously, they do not have a spine, or a backbone. All but one animal phylum are actually invertebrates, so there are many invertebrates. And so they lack an endoskeleton of bone or cartilage, is kind of their big characteristic to remember. Vertebrates, of course, do have a spine. They have an endoskeleton of bone or cartilage. Okay, they do have a backbone. Most of them are in the phylum Chordata. Okay, they're chordates, and we'll talk about them, at a later time. Now, here's the big evolutionary tree for invertebrates. These are all the big groups that we're going to talk about. Now, the bottom part is actually invertebrates up through echinoderms, and then chordates, most of them tend to be vertebrates also. Okay, so basically you had a common ancestor, and then you started to see tissues divide. Okay, and so the radial symmetry shows two tissue layers. Those are your cnidarians, like your jellyfish. If they have no tissues at all, that's really going to be your sponges. Okay, and then once they start to have three tissue layers, then that's when you get into your worms and your rotifers, and then you get into your arthropods like insects and your roundworms. And then finally, the most sophisticated are really the echinoderms, and then, of course, the chordates, which includes all the vertebrates as well. Now, organisms can be classified according to their 7 basic features, and here are the big 7. Okay, first off, what their symmetry is like. If they have no symmetry at all, if they have bilateral symmetry, if they have radial symmetry, we'll talk about that in just a few minutes. Body plan, how many body sections they have, the level of organization, that has to do with germ layers, and also what kinds of tissues that they have, what kinds of organ systems do they have. A coelom, it looks like coelom, but it's actually pronounced coelom. That's the body cavity of an animal. That's real important to know. The method by which a coelom is formed, or if it doesn't have any formation, that's an identifying characteristic in animal classification. So please keep that in mind. It's good to know. Segmentation, basically how they're divided up, that goes along with body plan and level of organization also. And then protostome or deuterostome development, that's really important too, and we'll talk about that just in the next few minutes. Some trends among animals that are good to know. The type of symmetry, it can be asymmetrical, which means it has no type of symmetry. The sponge that's blue here on the right side of the screen, that's an asymmetrical sponge. Okay, and a lot of sponges kind of have that. If it's radial symmetry, a lot of cnidarians like jellyfish, hydras, okay, even like an octopus, which is a cephalopod, they have circular organization, and so they can be cut in any plane to produce a mirror image. Okay. And then many animals have bilateral symmetry, which just means they have a left side and a right side, and if you cut down the midline, or that sagittal cut, that is going to produce a mirror image. Okay. And another characteristic of bilateral symmetry, it says here accompanied by cephali. That just means that they have a noticeable head region. So they have like a head and a brain and usually eyes. Okay, so they have a definite, top of their body. Now, the level of organization, sponges do not have true tissues. Okay, so they are considered really just to be kind of the same all the way throughout. Um, let's see, tissues. A lot of tissues have an ectoderm and an endoderm. Ecto means outside, and endo means, of course, inside. Cnidarians, like hydras, they have only like 2 tissue layers. And then the majority of animals have certain organs, they have an ectoderm, endoderm, and mesoderm that helps make up those 3 germ layers. And so they are actually making the animal have, definite organs. Now, embryonic development, animals with 3 tissue layers, they're either going to be protostomes or they're going to be deuterostomes or deuterostomes, however you want to say it. Now, protostomes and deuterostomes, they differentiate by how they divide or their cleavage, how their blastophore ends up, and then also how their coelom forms. And just for you to know, the deuterostomes, the echinoderms and the chordates, remember, are all your vertebrates. Echinoderms, those are like starfish, sand dollars, okay? And those tend to form in a very specific way. Likewise, the protostomes, those tend to be flatworms like tapeworms, your mollusks, your rotifers, arthropods like your insects. Okay? And so protostomes and deuterostomes, or deuterostomes, they divide and they form in a completely different way. Now, protostomes and deuterostomes, their cleavage is different. Now, cleavage is the first event in development, and that's basically cell division without a lot of cell growth. Okay. And in protostomes, the new cells are actually at an angle to the old cells when they start to divide. And so protostomes, they have cells that have limited potential and they can't really develop into a complete embryo. Okay, so the daughter cells are going to sit in grooves that are formed by previous cleavages. Okay, so you can see these little slits there on the side. That's cleavage right there. Those are cleavage slits. Now, deuterostomes, the new cells are going to sit on top of the old cells, and each one's going to be able to develop into a complete embryo. Okay, so some things that are good to remember. Proto in protostome means first, and then stoma actually means mouth. Okay, and deuterostomes, the deuter actually means second. Now, the meaning of those words actually becomes really important right on this slide. Now, basically, in protostomes, the mouth forms first, whereas in deuterostomes, the anus is going to form first. Okay, so protostome just means mouth first, basically. Okay, so please keep that in mind. In protostomes, the mouth is going to develop first, and in deuterostomes, the anus is going to form first. Then, in protostomes, the coelom, or the body cavity, is just going to have splitting through the mesoderm, or kind of that middle germ layer. And deuterostomes, you're going to have pouches that enlarge until they meet and fuse, and so basically their gut sort of out pockets, and they get their coelom, or their body cavity, that way. It's just a little bit more advanced in a deuterostome. Now, we're going to take the invertebrates sort of one big group at a time. The phylum Porifera, those are your sponges. Okay, basically you have kind of a sac-like body, okay, with lots and lots of little pores. They don't have nerve cells or muscle cells. They really lack organized tissues. They are multicellular. They are filter feeders, which are the very simplest kind of feeders. Basically, they filter their food through water by kind of a straining device. Okay, so they just strain out the food that they need and let the rest go. Okay, so they suck in water and get the food out that way. They reproduce by asexual and sexual reproduction. They can asexually bud, like yeast do, or some other, microorganisms. They can fragment and then grow themselves back that way. They can also sexually reproduce. They are hermaphrodites. Usually they don't self-fertilize. Okay, most of the time in sexual reproduction, the sperm is released through the opening of the sponge and then drawn into other sponges through the pores, and then they're going to fertilize eggs within the second sponge. So they basically kind of just transfer sperm sort of back and forth. Now, here's a picture of some sponges. Just so you know, sponges can regenerate. If the cells of a sponge get separated, they're able to reassemble and regenerate into a completely functioning organism, which is kind of neat. Now, the walls of a sponge here, they contain two layers of cells. You've got outer epidermal cells, and then you have inner collar cells. It's good to know that the collar cells have flagella they're going to beat and move the water through, and the food particles actually get trapped in these collar cells right here. Okay. And so then the sponge will transport nutrients just from cell to cell. Now, the next group, cnidarians, they have two germ layers, so they actually have true tissues, even though they're still pretty simple. They have kind of a tubular or bell shape. They have radial symmetry, which means that they're able to reach in all directions for their food. Most of them live in the ocean. Okay, all of them are aquatic, okay, mostly in salt water though. Now, they do have nematocysts used for prey capture. I'll show you a picture of that in just a second. They have two basic body forms. They have a polyp and a medusa. Okay, I'll show you that in a second too. And the cnidarians include your jellyfish, your Portuguese man-of-wars, your hydras, and then also your corals. Okay. Now, here's the life cycle of cnidarians. Basically, they have the mouth of a polyp that's directed upwards. Okay, if they're a polyp, and then the mouth of a medusa is going to be directed downwards. Okay. And some cnidarians actually have a polyp or a medusa stage where they constantly rotate through. And then in others, one stage may be dominant or absent altogether. Okay. Now, cnidarians actually have what's called nematocysts. Okay. And nematocysts, that's this long spiral coiled thread, and that's how they actually trap their prey. Okay. Here are some pictures of some cnidarians. You have sea anemones, you have cup coral, Portuguese man-of-wars, you have jellyfish right here. Coral reef ecosystems, they're actually very productive. They're extremely diverse. They have many, many groups of marine life that live there. Okay. And then you might have heard of Portuguese man-of-war. I always thought of them as like just really big jellyfish. They're actually a colony of modified polyps and medusa, and they tend to be very, very big, and they're very deadly. They will, of course, kill large animals. They have been known to kill humans if you get tangled up in them, but typically most humans don't run into them too much. Now, hydras. Hydras are cnidaria. They live in fresh water, okay, as opposed to a lot of other cnidarians, okay, that live in the ocean. They have polyps, so their opening is upwards. They have circular muscle fibers, a diffuse nerve net. They're very good at trapping prey. Okay, and they can reproduce sexually or asexually. Okay, they can sexually reproduce through ovaries or testes in the body wall, or they can bud themselves out asexually. They do not have medusa, okay, so their opening is always going upwards. Okay. And just so you know, because they have muscles and nerve fibers, cnidarians like this are capable of directional movement, so they can actually sort of move themselves even when there's currents wherever they need to go, wherever they need to find food. Here's what a hydra looks like. Okay. Now, when conditions are favorable, hydras will produce asexually, or they'll make buds like that and then pinch themselves off. Okay. They can pinch off and live independently. Okay. And then they can also sexually reproduce by the ovaries or testes in the body wall. Now, the body of a hydra contains two tissue layers. This is what it kind of looks like. You've got this gastrovascular cavity, and it's got a nerve net. Okay. And you've got some muscle fibers which help it move directionally wherever it wants to go. And then you also have these cnidocytes or nematocytes. These nematocytes close up are kind of scary looking. Basically, they have a trigger there, and if it gets tapped or touched, then the spines and the barb will come out and the filament will sting the prey. Okay. And that's how a lot of hydras and also cnidarians trap their prey. It's sort of like a poison dart. Okay, and when you have a lot of these, that's actually very paralyzing for their prey. Okay, so when it gets touched or tapped or moves up against something, then the barbs and spines and filaments will come out and they will sting their prey. Shoots out real fast.